Hello students, welcome to EPG Pathshala. I am Dr. Abhilasha Shauri from Manav Rachna International University, Faridabad. Today we are going to talk on module HPLC method development and validation under the paper Chromatographic techniques. After completing this module, you should be able to understand the importance of method development for HPLC analysis, to understand the process of method development in HPLC, and to learn about the process of validation of an HPLC method. Introduction to method development in HPLC. High pressure liquid chromatography is a column chromatographic technique employing high pressure pumps to pass both the mobile phase and the sample mixture through the stationary phase column and perform efficient separation. Analytical method development and validation are key elements of any development program. The HPLC analytical methods are required to establish the identity, purity, physical characteristics, and potency of the chemical, pharmaceutical compounds and drugs. Method development is largely done to support drug testing against specifications during manufacturing and quality release operations, as well as during long-term stability studies. Along with this, methods that support safety and characterization studies or evaluations of drug performance are also to be evaluated. Once a stabilized method is developed, the formulated drug product can then be subjected to heat and light in order to evaluate the potential degradation of the API in the presence of formulation. These are the components of the HPLC unit. HPLC method development. Method development is defined as the process of demonstrating that the method developed for the intended purpose or use is suitable and well optimized. The most appropriate HPLC method to be developed is selected depending upon the physical and chemical properties of an analyzed drug. Information regarding sample such as structure, molecular mass, pK values, UV spectrum and solubility of compound should be checked for. Also, the requirement of removal of insoluble impurities by filtration, centrifugation, dilution or concentration to control the concentration and extraction of the liquid or the solid phase, derivatization for detection, etc. should be thoroughly checked. For best mobile phase and columns for HPLC method development to be selected, the sample solubility should be determined as to whether the sample is soluble in organic or the aqueous solvent. Critical components for an HPLC method. The preliminary method development stage involves the investigation of all the components to be used in the entire process. This gives the analyst a chance of critical evaluation of each component and optimization of the final method. As time is a crucial factor, so time percentage spent on each stage is noted so as to enable the analyst to spend sufficient time for each stage. Selectivity can be manipulated by combination of different factors like solvent composition, type of stationary phase, mobile phase, pH of buffers, etc. Changing the solvents and stationary phases are the most comfortable approaches to achieve the separation. The proper range of pH is an important tool for separation of ionizable compounds. Acidic compounds are retained at low pH while basic compounds are mostly retained at higher pH. The neutral compounds remain unaffected. Steps for HPLC method development. For starting with the HPLC method development, following steps are required. Number 1, all the information on the sample should be gathered. The separation goals should be well defined. Special procedural requirements such as sample pre-treatment is to be sorted. The detector selection and setting has to be done. The separation conditions should be optimized. The problems or special procedural requirements should be checked for. The purified material should be recovered, and the calibrations for qualitative and quantitative methods should be done. Finally, before the release of the compound to the laboratories, method is to be validated. The requirements for good method development are column selection, selection of stationary phase material, mobile phase and its composition, selection of detector, and selection of pH range or polarity range of the mobile phase and the sample mixture. Column selection. The most commonly used HPLC columns are C18, although C8, the octyl bonded phases, are also used occasionally. The C18 are non-polar columns but not as hydrophobic. Therefore, retention time for them is higher. As for the hydrophobic compounds, it is typically shorter. Also, they may show somewhat different selectivity than C18 due to increased base silica exposure, unique selectivity that results in proton interaction of the bonded phase with electron deficient functional groups of solute molecules. Column dimensions. It refers to the length and internal diameter of the packing media bed within the column tube. On the basis of length, columns can be short columns ranging from 30 to 50 millimeters of length and long columns ranging from 250 to 300 millimeter of length. Short columns have short run time, fast equilibration, show back pressures and show high sensitivity. Conversely, the long columns have higher resolving power but offer more back pressure, have long analysis time and require more solvent. On the basis of internal diameter, columns can be narrower, ranging from 2.1 millimeter or smaller diameter, or they can be wider columns from 10 to 22 millimeter of diameter. Narrower columns inhibit sample diffusion. These are narrower and gives narrow and taller peaks and have lower limits of detection. Wider columns conversely have larger capacity. Column temperature. Separation of any sample can be enhanced by selecting the right column temperature. Higher column temperature reduces the system back pressure by decreasing the mobile phase viscosity, which in turn allow use of longer columns with higher separation efficiency. However, an overall loss of resolution between mixture components in many samples occurs by increasing the column temperature. The optimum temperature is dependent upon the nature of the mixture components. The overall separation can be improved by simultaneous changes into column temperatures and mobile phase composition. Stationary phase material selection. It refers to the properties of the packing media that forms the solid phase material in HPLC. Following considerations are included: number 1, particle shape; number 2, particle size; number 3, surface area of the particles; number 4, pore size of particles; number 5, bonding type; number 6, carbon load; and number 7, end capping. Particle shape varies from spherical to irregular. Spherical particles are more common in present-day analysis. Advantages of using spherical particles over irregular shaped ones is that they offer reduced back pressures and have longer column life in case of using viscous mobile phases, for example 50-50% of methanol and water. Particle size. It is the average diameter of the packing media particles. A standard range of particle size is from 3 micrometers to 15 to 20 microns. The smaller size particles give higher efficiency while larger particle range is used for preparative chromatography. Small particle size packaged columns are denser, allow less diffusion of sample bands between the particles and cause narrower and sharper peaks. However, smaller particles also cause higher solvent back pressures. The rule of thumb for selection of particle size and for resolving complex in multi-component samples is that normally 1.5 to 3 micron particle size are chosen. Surface area of particles. Total surface area of a particle is equal to the outer particle surface and the inner pore surface. The solute retention depends upon the surface area of the column bed particles expressed in square meter per gram. High surface area of particles show high solute retention, greater capacity and higher resolution. As a rule of thumb, a base material with maximum surface area is to be used for resolving complex and multi-component samples. Pore size. It is average size of the pores or cavities present in porous packing particles. The pore size range is 60 angstrom at the lower end up to 10,000 angstrom at the higher end. Larger pores allow high solute retention for maximum exposure to the surface area of the particles. Bonding type. Bonding type refers to attachment of bonded phase to the base material. There are two types of bondings, polymeric bonding and monomeric bonding. In polymeric bonding, the bonded phase is attached to base material via multi-point attachment. It offers increased column stability and enables high sample loading. In monomeric bonding, the bonded phase is attached to the base material via single point attachment. Carbon load. Carbon load refers to the amount of bonded phase attached to the base material. Good indicator of hydrophobic retention like in case of C18 or C8 and phenyl packings. Higher carbon loads gives higher column capacities, greater resolutions and longer run times. Low carbon load shows low run times, different selectivity because of greater exposure to the base material, and for maximum degree of separation, high carbon load columns are chosen. End capping. End capping is the process of bonding short hydrocarbon chains to free silanols remaining after the primary bonded phase has been added to the silica base. It is applied only to reversed phase chromatography. It reduces the peak tailing of the polar solutes that interact with mostly acidic silanols. Non-end capped packings provide a different selectivity than do the end capped packings, especially for the polar samples. Mobile phase composition. Mobile phase composition or solvent strength plays an important role in reverse phase HPLC separation. Acetonitrile, methanol and tetrahydrofuran are commonly used solvents in reverse phase HPLC having low UV cutoff of 190, 200 and 212 nanometers respectively. These solvents are miscible with water. Mixture of acetonitrile and water is the best initial choice for the mobile phase selection during the method development. pH range of mobile phase and sample mixture. The selection of pH range for better resolution between two or more peaks of an analyte depends upon three factors: column efficiency, selectivity, and retention time. Change in pH improves the retention time and thus can lead to easy separation of ionizable analytes from the non-ionized analytes. Change in the mobile phase pH can improve the column efficiency because it alters both the ionization of analytes as well as the residual silanols. It also minimizes the secondary interactions between the analytes and the silica surface that leads to poor peak shape. Optimum resolution is achieved by changing the pH of the mobile phase. Detectors. Various detectors used in the HPLC instrument include UV visible detector, photodiode array detector, fluorescence detector, conductivity detector, refractive index detector, electrochemical detector, mass spectrometer detector, evaporative light scattering detector. UV visible detectors are widely used in laboratories as they can detect a wide array of compounds. Method validation of HPLC. The validation of an analytical method demonstrates the scientific soundness of the measurement or characterization process. It is required to provide validated data throughout the regulatory submission process. The validation practice demonstrates that an analytical method measures the correct analyte in the correct amount and in the appropriate range for the intended samples. It allows the analyst to understand the behavior of the method and to establish the performance limits of the method. The goal is to identify the critical parameters and to establish acceptance criteria for method system suitability. Validation is defined by the International Organization for Standardization as verification where the specified requirements are adequate for an intended use, where the term verification is referred to as provision of objective evidence that a given item fulfills the specified requirements. The applicability and scope of an analytical method should be defined before starting the validation process. It includes defining the analytes, concentration range, description of equipment and procedures, validation level and criteria required. The validation range is defined by IUPAC as the interval of analyte concentration within which the method can be regarded as validated. Various validation parameters include linearity, accuracy, precision, richness, robustness, limits of detection, selectivity or specificity. Linearity. Linearity is the ability of an analytical procedure within a given range to obtain test results that are directly proportional to the concentration of the analyte in the sample. Essential to determine the useful range at which the instrumental response is proportional to the analyte concentration. Generally, a value of correlation coefficient R is greater than 0.998 is considered as the evidence of an acceptable feature of the data to the regression line. Significance of deviation of intercept of calibration line from the origin can be evaluated statistically by determining confidence limits of the intercept generally at 95% level of confidence. Accuracy. Accuracy is defined by ISO as closeness of agreement between a measured quantity value and a true quantity value. It is a qualitative characteristic that cannot be expressed as a numerical value. It has an inverse relation to both the random and systematic errors where high accuracy means lower errors. Accuracy is evaluated by analyzing test drug at different concentration levels. Precision. Precision expresses closeness of agreement or a degree of scatter between a series of measurements obtained from multiple sampling of the same homogeneous sample under the prescribed conditions. Precision may be considered at three levels: repeatability, intermediate precision, and reproducibility. Repeatability is also referred to as an intra-assay precision. It is the measure of precision of analysis in one laboratory by one analyst using one piece of equipment over a relatively short span of time. It is degree of agreement of results when experimental conditions are maintained as constant as possible and expressed as RSD of replicate values. ICH recommends a minimum of 9 determinations covering the specified range for the procedure. Example, 3 concentrations per 3 replicates as in the accuracy experiment, or minimum of 6 determinations at 100% of the test concentration for evaluation of repeatability, which should be reported as standard deviation, relative standard deviation, or coefficient of variation, or confidence interval. ICH defines the intermediate precision as the long-term variability of the measurement process and is determined by comparing the results of a method run within a single laboratory over a number of weeks. It is also called an intraday precision. It reflects discrepancies in results obtained by different operators from different instruments with standards and reagents from different suppliers with columns from different batches or a combination of these but in the same laboratory. Intermediate precision is also refers to the verification of the method that will provide same results in the same laboratory once the development phase is over. Reproducibility expresses precision of analysis of the same sample by different analysts in different laboratories using operational and environmental conditions that may differ but are still within the specified parameters of the method. The objective is to verify that the method will provide the same results despite differences in the room temperature, humidity, variedly experienced analysts, different characteristics of equipments such as delay volume of an HPLC system, variation in material and instrument conditions such as in HPLC, the mobile phase composition, pH and the flow rate, equipments and consumables of different ages, columns of different suppliers or different batches, and the solvent, chemical reagents and other materials with different quality. Selectivity and specificity. Selectivity and specificity are sometimes used interchangeably to describe the same concept in method validation. Selectivity of an analytical method is defined by the ISO as property of a measuring system used with a specified measurement procedure, whereby it provides measured quantity values for one or more measured, such as the values of each measurement are independent of other values or quantities in the phenomena. Specificity is the ability to assess unequivocally the analyte in the presence of components that may be expected to be present. The specificity of a test method is determined by comparing the test results from an analysis of sample containing impurities, degradation products, or placebo ingredients with those obtained from the analysis of samples without impurities, degradation products, or placebo ingredients. Specificity can best be demonstrated by resolution between the analyte peak and other closely eluting peaks. Detection limits and quantitation limit. Limit of detection of an analytical procedure is the lowest concentration of an analyte in a sample which can be detected but not necessarily quantified as an exact value. Limit of quantitation is the least amount of analyte in a sample which can be quantitatively determined with suitable precision and accuracy. Three methods are there for determining LOD and LOQ according to the ICH guidelines. These methods are, firstly, the visual evaluation. Visual evaluation may be used for both non-instrumental and instrumental methods. The LOD and LOQ is determined by analysis of sample with known concentrations of analyte and by establishing the minimum level at which the analyte can be reliably detected or quantified with acceptable accuracy and precision, respectively. Signal to noise ratio approach. This method can only be applied to the analytical procedures which exhibit baseline noise. It is determined by comparing the measured signals from samples of known low concentrations of analyte with those of blank samples and establishing minimum concentration at which the analyte can be reliably detected. A signal-to-noise ratio of 3:1 is considered acceptable for estimating LOD with relative standard deviation less than or equal to 10%. Limit of quantitation, a signal-to-noise ratio of 10:1 is considered appropriate with relative standard deviation of 3%. Limit of detection and limit of quantitation via signal-to-noise ratio may be expressed by a certain formula in which the slope of the calibration curve may be estimated for an analyte. The value of the sigma or the standard deviation may be taken from the analytical background responses of an appropriate number of blank samples. Alternatively, it can be taken as the residual standard deviation of a regression line or standard deviation of y-intercepts if regression lines are obtained for samples containing an analyte in the range of LOD and LOQ. Range. The range of an analytical procedure is the interval between the upper and lower concentrations of analyte in the sample, for which it has been demonstrated that the analytical procedure has a suitable level of precision, accuracy, and linearity. It is established by confirming that the analytical procedure provides an acceptable degree of linearity, accuracy, and precision when applied to samples containing amounts of analytes within or at the extremes of a specified range of the analytical procedure. The range of an analytical method varies with its intended purpose. It can be 80 to 120% of the test concentration for an assay of a drug substance or a finished drug product. It can be 70 to 130% of the test concentration of content uniformity plus minus 20% over the specified range of dissolution testing. It can be lesser if the impurities are to be quantified. It should be, however, commensurate with LOD or LOQ, the control level of impurities, for impurities known to be unusually potent or to produce toxic or unexpected pharmacological effects. Robustness. Robustness is the measure of the capacity of the procedure to remain unaffected by small but deliberate variations in method parameters and provides an indication of its reliability during its normal usage. For determination of method robustness, a number of chromatographic parameters are used. For example, flow rate of mobile phase, column temperature, injection volume, detection wavelength, mobile phase composition. All these parameters can be varied within a realistic range and quantitative influence of the variables is determined. So students, let us now summarize what we have learned in this module. Method development and validation are key elements for any analytical process. It is essential to establish the identity, purity, physical characteristics, and potency of the chemicals, pharmaceutical compounds and drugs. It supports the drug testing against specifications during the manufacturing, their quality assurance and stability. Validation proves the scientific soundness of the process, measurement and characterization. Thank you.